This first slide is simply taken from your textbook, giving you an idea about how much energy comes from aerobic respiration, as you will see in this chapter. In the glycolysis and citric acid cycle, you have seen reactions that produce NADH and QH2, and this is also true for fatty acid oxidation as well as for amino acid oxidation, as you will come to learn later. Now, these NADH and the QH2 are reduced coenzymes that are rich in energy, as you will see in the next slide, and they will be used in the production of ATP. ATP in living cells can be produced by substrate level phosphorylation, as you learned in glycolysis and citric acid cycle, by oxidative phosphorylation, which is the topic of this lecture, and this happens in the inner mitochondrial membrane, as well as in photosynthetic photophosphorylation, which occurs in the thylakoids of the chloroplast. I won't be able to cover the photosynthetic phosphorylation because I don't cover photosynthesis in this course. How is NADH and QH2? How is it oxidized in the inner membrane of the mitochondrion? This is done through a series of protein complexes that are located in the inner membrane of the mitochondrion. They are known as complex 1, 2, 3, and 4. And the electrons will pass from NADH through these complexes, eventually through a series of electron carriers, as you will learn in the coming slides, and they will at the end reduce oxygen to water. And this is true for QH2, where the QH2 is oxidized to Q, again passing through a series of electron carriers to reduce oxygen to form water. There is another complex in the inner membrane. That complex is called ATP synthase, and that complex is involved in the synthesis of ATP. Before I start discussing the electron transport chain in the inner membrane of the mitochondrion and oxidative phosphorylation, let me go briefly over the structure of the mitochondrion, which you are aware of. Here, in the figures in front of you, there is a schematic representation of the mitochondria showing the different compartments that make up the mitochondrion, and these are the outer membrane, the inner membrane. The space between the outer membrane and the inner membrane is called intermembrane space, and the space that is surrounded by the inner membrane is called the matrix. And as you see, the inner membrane is forming finger-like projections to the inside. It is highly folded to form what we call the cristae, and that will increase the surface area of the membrane, making it more efficient. On the same slide, there is an electron microscopy section showing how the mitochondria looks under the electron microscope. Okay. The outer membrane, as I told you earlier, is a smooth membrane. It's highly permeable. It can allow substances up to 10,000 daltons to pass through it. While the inner mitochondrial membrane is a semi-permeable, highly selective membrane. It's excessively folded, forming the cristae. Proteins contains like 75 to 80% proteins. It's very permeable to oxygen, water, carbon dioxide. It contains the components of the electron transport chain as well as ATP synthase and many carrier proteins. The intermembrane space is usually a continuation of the cytosol because, as I said, the outer membrane is permeable to many substances, so they can cross easily from the cytosol to the intermembrane space. The matrix is a gel-like substance of about 50% water. It has a lot of enzymes, like pyruvate dehydrogenase complex, the enzymes of the citric acid cycle, with the exception of succinate dehydrogenase complex, which is located in the inner membrane of the mitochondrion, as we learned earlier. The enzymes for oxidation of fatty acids, phosphoenolpyruvate carboxykinase, important enzyme in gluconeogenesis. I'll come to discuss that later. Many coenzymes like NAD+, NADP+, as well as DNA, ribosomes, and the machinery for protein synthesis. Before I discuss the different complexes that make up the electron transport chain, let me go over the classes of the electron transport chain compounds that are involved in the electron transport. And there are four compounds associated with the electron transport chain. Pyridine dehydrogenases, they contain NAD plus or NADP plus as coenzymes, and I discussed earlier how NAD plus or NADP plus are involved in carrying electrons. Flavin and dehydrogenases, they have alpha and again I explained earlier when I discussed coenzymes, I explained how FMN and FAD are electron carriers. Another compound is called coenzyme Q or ubiquinone, which I'll be discussing in the next slide. And there are protein coenzymes, two classes, cytochromes and iron receptor proteins. The ubiquinone is found in most organisms. That's why it's called ubiquinone from the English word ubiquitous, meaning it's present everywhere. It's benzoquinone, it has four substituents. One of these substituents is a very large tail made up of something like 6 to 10 5-carbon isoprene isoprenoic bonds. Ubiquinone can be present, as you see, in three forms, either fully oxidized Q, partially reduced semiquinone, or fully reduced QH2 or ubiquinone. And it can be reduced in two 1-electron steps via the stable free radical semiquinone. So ubiquinone can receive 1 electron to be reduced to semiquinone, then another electron with 2 protons to receive to be reduced to QH2. QH2 can donate 1 electron at a time. So it functions in a way similar to FMN and FAD, which I explained earlier. The reactive center of the coenzyme is shown here.